Hello and welcome to the Chemistry Topic Mixtures, Lesson 3, Separating Techniques, Part 2. So in the previous lesson, we looked at filtration and evaporation. We're going to look at two slightly different techniques this time. The first being distillation. This will be one that you will see happen. You won't necessarily get to do it yourself. It will be done as a demo. Click on the video and watch it because it will show distillation taking place. Okay. So distillation is one, and then the other one is chromatography. And chromatography can be done in two ways, which we will talk about in a minute. So remembering that we're still talking about separating a mixture, not a compound. So for distillation, in our video there, we've got a mixture of ink and water. And what we want at the end of the process of distillation is pure water. So we will heat the solution until it boils, and then what you will see is the water evaporating, and the steam will collect at the top, and then it will be passed through a condenser. The condenser tube is where that arrow is at the moment. And you can see the orange tube sort of wrapped around that. That will be attached to a water source, and it'll be attached to a cold water source because as the steam passes through the condenser, it is cooled. Okay, so condensation will happen, and what you will then get is condensed water, which is pure, and that's collected in a flat-bottomed conical flask at the bottom. So you go from having a coloured ink solution to pure water. It's a really good visual experiment to give you an idea of distillation. And you've got to remember that you've got boiling, evaporation, and condensation taking place in distillation. So the second method we'll look at is chromatography, and this is used to separate dyes in inks. There are two methods to do this. This first method is the one where you can look at one or more ink colours at once. So on the picture there, they're just looking at one, and you can see here that they've drawn a tiny pencil line. They've spotted originally the ink here, and then they've placed their chromatography paper in the solvent, and that solvent has spread, taking the inks and separating them out up the chromatography paper. And it shows you the different dyes that were present in the original colour inked. Now, the other method is one that uses round chromatography paper, and a wick is cut so that it points down. Now, when I click onto the next slide, hopefully you'll be able to see a video of this. There won't be any sound, so I'll talk through it. Okay, so this one's looking at black ink, and you've put the black dot on the filter paper. You cut it to make the wick, and then you'll place that in a beaker of water, the water acting as the solvent, and there you go. You can see them separate out, and you can see the different colours there that make up black ink. That can be tried with all sorts of different colours, different coloured pens. Okay, it's a good one to look at the dyes that make up the different inks. So one on a straight piece of paper that you can put several colours on, and one using that example that we've just seen there. Now remember that uses of chromatography, two really important uses of chromatography are identifying blood samples and also investigating chlorophyll. Now chlorophyll is the chemical in leaves that's involved in photosynthesis. Okay, finally, we just need to check whether our substances that we've then separated are actually pure. So they could be pure or impure. Now, the way you can work this out, if the substance is pure, it has a fixed melting and boiling point. So the melting point is the temperature at which that solid becomes a liquid, and the boiling point is the temperature at which the liquid becomes a gas. So those will be fixed if the substance is pure. Now, if the substance is impure, it will have a different boiling point. So for example, if your liquid boils at 100 degrees, it is very, very likely it is water, and pure water. If it boils at a different temperature, then you're looking at still a slightly impure substance. There's something else present. So those are the points to remember for pure and impure substances. Okay, they're the last two separating techniques we need to talk about, distillation and chromatography. That means we've covered 4 in the last 2 lessons, and we've also just done a little bit of detail on what makes a pure and impure substance. So you need to go off and complete the final worksheet with this topic, and then it will be time for you to do the quiz and check your checklist, and we will see you again very soon.